Thank you very much, Sharat and Team Genesys, as well as Team NIT Raurkela. Thank you very much for coordinating this particular workshop. So it's all our pleasure to conduct workshop at such a nice place, NIT Raurkela, with all the really talented students from your institute. And I again specially thanks to all dignitaries who have participated in the inauguration ceremony and shared their valuable inputs. And I hope those inputs will be really helpful for all of the participants. And onwards, you will definitely see throughout this workshop, like how one could use the computer-aided drug designing technology for designing really novel or new potent drugs. So this is what the aim of this workshop. But so before starting this workshop, we always encourage to know little bit background of all of you, that how you are placed in terms of computer-aided drug designing or molecular modeling studies. So for that, I just share with you a small poll. So here you go. So whoever using Zoom app, the poll must be popped up in front of you. So it's just a simple three questions which you need to answer. It's all anonymous poll. Please try to answer the poll question. It just help us to understand in better way, like what is your experience and how what is your understanding about that field. And then it will help us to tweak our presentations and our further sessions accordingly. So please try to participate in the poll. Thank you. Yeah, it's good to see like most of the participants are participating in poll and it's overwhelming response. Thank you very much for it. So I just wait for 30 more seconds and then I'll close the poll and we'll have a small discussion on it. So please try to answer the poll questions if it is not yet done. Thank you. Yep. So here you go. I just end the poll as most of you have already participated in the it. So just end the poll and I'll be happy to share the results with you, so that you also see what is the distribution of all of you. So here you go. The poll results must be in front of you. So it's all anonymous poll, so nobody knows the name, so don't worry about it. So we'll just see the distribution of the questions which we asked for. So first question was, please share your experience or exposure to computational chemistry or computational biology. And as expected, most of you are the freshers to the field. So that's right, really fine. Some of you are also in the basics or medium, or few of you are also advanced. So that's great. We will try to cover it from really basic so that you just start understanding the field from ABC. And this is how we will tweak our presentation a little bit. And then second question was, did you have earlier experience with cloud or cloud-based workshop? And most of you said no. So welcome, welcome for this particular cloud-based workshop. And this will be really your first experience. That will be really great. I hope so. And we will be there to support you in any case, any queries if you have, if you feel any difficulty in working with cloud. So we are always there to support us. Please be with us and try to do the task as mentioned by our team. Third question was a broad field of your expertise, and it's really good distribution. Many of you are from biology background, few of you are from chemistry background, and many of you are also from technology and others. So the biology and chemistry is the field which is of our interest, but it's not again limited. It's all interdisciplinary. The student from technology background, you will have a lot of things to learn, and this is how we have planned this workshop for, so that it gives overall glance for all kind of interdisciplinary students. So thank you very much for participating in the poll. So I just stop sharing the results. And now we can proceed for the actual workshop. So before proceeding for the actual workshop, it is always good to know about the field, to know the basics of field. And for that, I would like to welcome our principal scientist, Dr. Pritesh Bhatt, who will take you through the field and will explain all the things in detail. And for that, I welcome him. And just to mention one more thing, so I request all of the participants to please keep your mic on mute mode, so that there should not be any disturbance during presentation is going on. And on the top of that, if you have any questions, any queries, as pointed out by Raghu in the talk, you can always reach out to us. So please post your question in the Zoom chat box, or we will have enough time after the presentation to take all of your queries. So if you want to talk, we can have discussion after the presentation, or in meantime, just post your question in the chat box. We will not leave any stone unturned. We will take all the questions and answer your all the questions. So thank you very much again, and over to you, Dr. Pritesh.
Thanks, Prajwal. Can you please confirm if you can see my screen and my voice is clear?
Perfect.
Thank you. Right. So as Prajwal mentioned, so in this presentation, I'll be touching upon very basic thing. Okay, so we'll try to understand what is mean by disease, what is mean by drug design, what is mean by computational drug design. I hope you are able to understand. Drug design is different, computational drug design is different, right? So what is the logics involved in when we say computational drug design? All those things we will try to understand. Again, if you have any question, please post it in the chat room. At the end of the presentation, we will take up the question and we'll have a discussion on that. Right. Okay. So what is the necessity for going uh, uh, for drug discovery? So drug discovery is required. Okay, before we go into the drug discovery, let us try to I'll give you a brief background about Schrodinger software. So we have different suites, different types of modules. I am sure you might have used PyMol. This is a fantastic visualizer, nice pictures you can generate, nice visualizer you have here. Then we also have a suite called as Bioluminate, which is specific for biologics design, so proteinaceous drug molecule design. So here we have tools for designing vaccines, antibodies, protein engineering tools, protein liability prediction tools. All those things we have in the biologics module. So in today's workshop, we'll be discussing majorly aspects from small molecule drug discovery suite. So here we come across docking, QSAR, machine learning model, molecular dynamics, homology modeling. So these type of aspects we cover. We also have material science suite, that is for predicting materials property. So let us go into drug discovery, right? So why is drug discovery needed? It is to treat a disease, right? So what is mean by disease? Let us try to understand. So it is an abnormal condition. So what is that thing which maintains normal condition or body homeostasis? It is the proteins which maintain the normal condition. So if something goes wrong with the protein, that results in disease, right? So don't get confused between disease and infection. Infection is the invasion by external organism, bacteria, viral invasion, right? So that is infection. So I am talking about the disease here. Okay. So now we understand there is something wrong going with the protein. So what type of abnormality you can have? So there could be either overactivity or underactivity of the protein, right? So what could be the reason for these altered activity of the protein? So the first direct, very simple to understand, the first situation is altered quantity of synthesis, right? So number of molecules protein required. Let us imagine 50 molecules of enzyme is required. So instead of having 50 protein molecule, now only 20 are there. Or instead of having 50, now 70 are there. So now you can easily understand. So if more quantity of protein is synthesized, then the output also increases. So that would result in overactivity. The second scenario is the number of molecule of protein is exactly same. There is no change in there, but there is altered altered activity in the protein. So the catalytic activity has changed in the protein. How that could happen? That is because of the mutation, right? So these proteins are mainly involved in bringing some chemical modification, chemistry changes they bring, right? Catalysis they do. So during catalysis, there would be a transition state. So there would be an energy barrier which a product has to cross to form the substrate has to cross the energy barrier to form the product. So during with the mutations, with the change in the amino acid sequence, these transition state energy barrier may come down so that, or it may increase also. If it comes down, the product will form very easily, right? So then that may result in overactivity. If the energy barrier increases, that means lesser product will form. So in all these cases, there is altered activity in the protein. So since we are talking about protein, let us try to understand. You know all these aspects. I'm just brushing up here about the information. Protein is a polymer. It is made up of amino acid residues, and 20 different types of amino acid residues are there. You can class them depending on their nature. There are hydrophobic amino acid residues, right? Alanine, valine, leucine, isoleucine. There are aromatic amino acid residues, tryptophan, tyrosine, phenylalanine. There are charged amino acid residue, positive and negative charge, right? So polar amino acid residue. So these amino acid residue form a polymeric structure, right? So the amino acid sequence information, we call it as primary structure, right? So these will have a particular folding pattern that they form a hydrogen bond network, and they take a particular folding pattern. So that is what we call it as secondary structure, helix, beta pleated sheet, right? The secondary structure, they form a globular shape, that is called as tertiary structure. And several these type of tertiary structure might come together, we call it as quaternary structure. So it is the tertiary and the quaternary structure which is very important for exhibiting activity. So if you heat the protein, if protein loses this tertiary structure, or in other words, if protein gets denatured, it loses its activity. So always remember, it is a tertiary structure which is very important for exhibiting protein activity. So again, you might have heard about proteins. These could be enzymes or receptors, right? There are two types of proteins what you can find in the body. So what is meant by enzyme? So earlier I mentioned proteins bring out, these are the ones which bring out chemical modification. So the proteins which are involved in chemical modification, okay, so those are called as enzymes. So cell wall component synthesizing enzymes, or RNA or DNA component synthesizing enzymes, right? So these are the ones which bring out chemistry modification, or the enzyme which is involved in the digestion. So these bring out chemical modification. So these are the enzymes. But proteins could be also receptors. So receptors are involved in signal transduction. Okay. So whether it is enzymes or the receptor, they can exist in two different forms, active and inactive form. Okay. So in the active form, an enzyme will be able to bring a catalysis. A receptor would be able to propagate a positive downstream signaling. Okay. So in the inactive form, enzyme will not be able to bring any catalysis, whereas in the case of receptor, it will propagate negative downstream signaling. Okay. Now it is very important to understand the difference in these two forms. Okay. What is mean by active form of protein? What is mean by inactive form of protein? And what is the difference? Okay. Does amino acid sequence change? No, primary sequence remains same, right? So is there any chemical changes happening in the protein? No, because amino acid sequence remains same. So what is in it makes or brings this difference? In one form, it is able to bring the catalysis, and in other form, it is not able to bring the catalysis. Okay. The main difference here is change in the shape. Okay. Change in the shape. That's all. Okay. In one shape, the protein is able to bring the catalysis. We call it as active form. And in other shape, the protein is not able to bring the catalysis. We call it as inactive form. Similarly, in case of receptor, in one shape, it propagates positive signal. In other shape, it won't be able to propagate positive signal, right? So how this shape change is triggered? Okay, when the substrate binds. So you can see here in the graphics, it's an imaginary picture. You can see a pink color molecule when binds to the protein, or a substrate when binds to a protein, it induces the shape change in the protein. Okay, so from one shape to other shape it will induce, right? So now when we are talking about substrate, like for example adenosine triphosphate, ATP. Okay, ATP is the substrate for kinase. So when we talk about substrate, this is a very special molecule, because substrate will have very special type of property in it. The first property is affinity. The moment you put it with the protein, it loves to bind with the protein. It has a very strong friendship with the enzyme. The moment you put it, it just goes and gets itself bound or attached to the protein. Okay, and the second special property is intrinsic activity. This is very special now. Okay, affinity is special, intrinsic activity is very, very special. What is the speciality? When the substrate goes and binds to the protein, it induces this shape change. Now normal molecule will not be able to induce this type of shape change. Only substrate, okay, that special molecule only would be able to induce this type of shape change, and that is called as intrinsic activity. Okay, so what is the driving force for the affinity? So I said the moment you put the molecule with the protein, it just goes and binds to the protein, right? There, what is the driving force for this affinity? The driving force for the affinity is non-bonded interactions. Okay, what is meant by that? There is no covalent bond formation. Okay, there is no permanent bond formation. These are weak type of interactions. Hydrogen bond, hydrophobic interaction, dipole-dipole interaction, right? So these type of interactions. The speciality of these interactions are is they exist in multiple number. It's not the only one hydrogen bond will be there or something like that. They exist in multiple number, and they will exhibit a synergistic effect. It's not the summation effect. 1 plus 1 will not become 2 or 3. It will become more than that. It will have a synergistic effect. 1 plus 1 could become 4, 5, 6, right? So even though their individual contribution is very weak, since they exhibit a synergistic effect, they will exhibit a very strong effect when they go and bind to the protein. Okay, so that is why always you keep in mind when you're analyzing the interaction between small molecule with the protein, you have to look for all the type of interactions. Don't think that only the hydrogen bond is important, only hydrophobic interaction. No, all the type of interactions are equally important because they will exhibit synergistic effect. Okay, now let us try to understand what happens in the disease condition. So we already discussed, right? So let us imagine there is overactivity. Okay, either because of mutation or because of increased quantity, any uh, let us assume any of these factors. Let us assume there is an overactivity. So substrate is available in abundance. So the moment it goes and binds, it would further trigger the activation of the protein and disease condition aggravates, right? So to control the disease, you have to control the activity of the protein, right? So how do you do that? So you have to prevent the conformational change or the shape change happening in the protein. If you stop that, okay, this is specifically in case of enzyme. Okay, so if you prevent the shape change happening in the protein, then you can suppress the activity of the protein. Okay, so this is specifically for enzyme. How can I do that? So the first approach is competitive inhibitor. Wherever the substrate is going and binding to the same place, you bring in your own molecule. Okay, it could be a synthetic molecule which you can synthesize in the lab, or it could be from phytochemical origin, plant origin, right? So now if your molecule has to bind, so what we are trying to do, we are exploring whether our molecule can it go and bind to the place where the substrate is binding. Now keep in mind the substrate is always present in the body and it is present in abundance. Whatever medicine you take, very short time you are taking, very small quantity you are taking, in that concentration it should be able to exhibit activity, right? 2 days, 3 days you are taking 5 mg, 1 mg, 0.5 mg tablet or injection, you take it, right? So what I mentioned here, what is the driving force? The driving force is the affinity, right? Non-bonded interaction. So if our designed molecule is able to exhibit higher binding affinity than the substrate, then our molecule would preferentially come and occupy the space. Now our molecule will have the special property of affinity, but it doesn't have the intrinsic activity. So it would come and bind, but it won't be able to induce this type of shape change, right? So once protein, since the space is already occupied by our molecule, now substrate won't be able to come and occupy the space. If substrate doesn't bind, protein doesn't change the shape, and now protein won't be able to exhibit any activity. So it is stuck with a particular shape, it loses its activity. So there is a competition between substrate and our designed molecule. So that is why we call it as competitive inhibitor. The alternate approach is you explore some other place than the substrate binding site. We call it as allosteric site. Okay. You come up with a molecule which can bind to some other place than the substrate binding site. So when our designed molecule binds to the protein, you can see it has altered the shape of protein. You see here, so the binding pocket is wide and open so that the substrate would be able to come and bind and occupy the space easily. But when inhibitor molecule comes and binds, you can see now it has altered the binding pocket. So now the binding pocket has become small, right? So it has altered the shape. With this altered shape, the substrate is not able to bind to the protein. If substrate doesn't bind, protein won't be able to uh, uh, induce the shape change. So if protein doesn't change continuously shape, it doesn't exhibit activity. So whether it is competitive inhibition or allosteric inhibition, ultimately you are preventing the shape change happening in the protein, and that is how you suppress the activity. Once again, this is very specific to the enzyme. Okay. In case of enzyme, if you just prevent the shape change, then the protein activity stops. In case of receptor, it is the shape which is very important. Okay. So if you want to design a agonist, okay, if you want to design an agonist, you have to block the protein in the activated form. In the activated shape, you have to block. If you want to design an inhibitor, you have to block the protein in the inactive form. So that is how you come up with the design of the molecule. So drug discovery phase is a long, it's a time consuming affair. Okay, it takes minimum 13 years and it's very expensive also, $2 billion US dollars it would take. Okay, so here I have listed few of the steps which are involved in drug discovery phase. It all starts with the target identification. So this is the first step where you try to identify a protein. If you modulate that protein, either inhibit or activate, whatever, depending on the requirement, if you modulate the activity of that protein, would exhibit required therapeutic activity. That is what you are confirming. That is called as target validation. So you do a knockout study, you inhibit that protein. So all those various techniques you use to confirm that the particular protein is of importance. Then once you identify that, then the first step is hit identification. So in this phase, you try to recognize at least a molecule or the chemical scaffold which can at least bind to the protein. So you may not expect very high activity for that molecule, but at least it would be able to bind to the protein. The next step is hit to lead. Okay. In this stage, you try to improve the potency of molecule. Okay. In the next stage, okay, lead optimization. You have here a highly potent molecule, very efficient molecule, but it might have some side effects. So you want to remove those toxic effects. So that phase is called as lead optimization. So all these molecules you have to first test in the preclinicals. So you directly don't test it in humans, right? So you do preclinical testing. So in vitro and in vivo testing. So first you isolate the enzyme in test tubes. You will confirm that the molecule is really effective. Then you try it on animals, mouse, right? So in vivo testing, we call it as. Once you confirm the molecule is really safe, okay, the first thing is whether it is really safe, whether it is really exhibiting activity. Once you confirm, then you approach the regulatory authorities with the data. In animals, in the preclinical stage, we are able to see the activity, give us permission for starting the human trials. That is what is the first stage. Okay. So a very small number of people it will be tested, maybe 10 or 15 or 20 people. That is phase 1 trial. Then larger population it will be tested. That is phase 2. Then in the third stage, still very large population it will be tested. So here in this stage they will confirm whether the molecule is really able to exhibit activity. Is it showing any toxic effects? So all those things they'll try to get the information. And once they prove once they see that the data is really promising, molecule is really good, then again they will approach the regulatory authority, FDA. Okay, and then they will show the data and they'll ask the permission for marketing the molecule. Now, why it is really taking so much time? I said 13 years it is taking and it's becoming so expensive, right? We understood now what is meant by drug discovery. So is it just sufficient if molecule binds to the protein? If molecule exhibit potency, is that sufficient? No, that is not just sufficient. Molecule should not have any toxicity, right? So it should be highly selective, it should have decent solubility, it should have decent bioavailability, clearance, permeability, it should have great synthetic feasibility, right? So now you are trying to identify one molecule having several properties in it, and this is what makes it complicated. So to identify one such molecule having several property, you have to really try and test. You have to synthesize and you have to test for all these property, right? So that is why it is really taking time, right? So in commercial scale, if you see, there would be separate chemistry team who would be doing the synthesis. There would be separate in vitro team. There would be separate in vivo team. There would be, you have to maintain animal facilities. You have to maintain the machineries. You need to have technicians. You need to have scientists, right? So all these things would come up, add up to the cost. So that is why it is becoming very expensive. So it is, I'm sorry, I missed to add the reference here. It was reported in Nature Review in publication that a commercial company would at least synthesize about 2000 molecules per research project. Out of 2000 molecules synthesized, it's only 10% of molecule would exhibit high potency, single digit nanomole concentration. Okay. So that means 10% of 2000 would be about 200 molecules. So out of 200 molecule, it's only 0.41% of molecule will have decent ADME profile. Okay, druggable profile. What is meant by ADME? Absorption, distribution, metabolism, and excretion plus toxicity. Okay, so druggable profile. So 0.41, if this becomes 0.5 out of 200 molecule, you can say one molecule. Now this is less than one molecule. You imagine how much risk is involved, right? So from 2000 molecule, you get only one molecule which could be druggable, and that one molecule becoming successful in clinical trial is only one in three molecule. So this is the statistics. This is the clinical trial statistics shows. Okay, and you can see how much it's really expensive. More than $800 million one has to spend, even if you just consider clinical studies, right? Is there a smart approach? Can you cut down the money involved? Can you cut down the time required? Is there any smart approach we can implement? Yes. So computational tools will help you. So let us try to understand what is meant by computational drug discovery. So how does the computer or computational tools can help in drug discovery? So now you understood what is meant by drug discovery. So ultimately I have to prevent the protein activity. So using computer, you try to find out a molecule which can prevent the proteins conformational change. Okay, so two approaches are there. The first approach is called a structure-based drug discovery. So here you utilize the information of the structure of the protein. Okay, target structure you would use. So you know where is the binding site, right? Whether it is allosteric site. There are two approaches, right? Competitive inhibitor or allosteric inhibitor. So wherever the molecule is binding, you get that information. We know protein is made up of amino acid residue, and amino acid residues could be of different nature. We just discussed in the first slide, right? Hydrophobic, there could be polar, there could be charged residue. So you explore what type of residues are forming the binding pocket. So accordingly, if you complement it, then they would be able to offer that type of interactions, right? So if you understand, if you see here, there is a aromatic amino acid residue in the binding pocket. That means that residue can offer pi-pi interaction. If there is a hydrogen bond donor group like serine or threonine, if it is there, it can offer a hydrogen bond. So in your molecule, you have to put a complementing thing. So you have to, if there is a donor group, then in your ligand, you should put, in your molecule, you should put an acceptor group so that it can engage those residues, right? So that is how you make utilization of the information of the binding site to bring or come up with the design of molecule. So that is, since you are making use of structural information of the target, it is called a structure-based drug discovery. So here we come across molecular docking technique, homology modeling technique, molecular dynamics technique, like so these type of applications, free energy perturbation. So these type of tools we use here. The second approach is ligand-based drug discovery. So now crystallizing protein is very difficult because it's a long polymer. Okay, polymer crystallization is very tough challenge. Okay, small molecule easily you make a heated super saturated solution, you cool it down, you get beautiful crystals. But polymer crystallization is very difficult. So many of the targets we may not be having structural information, right? So in that case, what we will do, already reported ligands information will take. Now, a lot of medicinal chemists would be there. They will use their own intuition. They'll go for the synthesis and they'll report this molecule was active, this was intermediate active, this was less active, this was inactive. We will use that information. And from there, we come up with the hypothesis. So here we use different techniques like pharmacophore, QSAR, artificial intelligence, machine learning model, shape-based search, right? So these type of technique we use in ligand-based drug discovery. Okay. So let us try to understand more about structure-based drug discovery. What is mean by that? So whenever a ligand is binding to the protein, we show like this, right? Lock and key model, right? So this is a very simple representation, but you understand it involves a lot of detailing also. See, if one of the teeth in the key breaks, okay, lock will not open, right? The same thing applies even here. Okay. So if a molecule, whether it is a substrate or an inhibitor, if it has to bind to the protein, it has to perfectly complement. Okay. So there are two types of complementarity expected if molecule has to bind to the protein. The first thing is geometric complementarity, shape complementarity. There should be a perfect fit. So here you can see it has two grooves. This is a protein. The green color stuff is protein. So there are two grooves. So in the ligand also we have introduced two grooves, right? So if this was round, still it would have occupied. Now keep in mind, in the body, everywhere water is there. You have seen that Madhuri Dixit ad, right? 70% body content is water, right? So everywhere, there is no vacuum inside the body. Everywhere water is there. And water is the greatest flusher. It can wash out anything, right? So if the ligand is not perfectly complementing, the water will get leaked inside and it will wash out. So the ligand would not be able to very effectively bind to the protein, right? The second thing, that is the shape complementarity. Okay. The second thing is electrostatic complementarity, charge complementarity. We already know the amino acid residues are of different nature, right? So it has to complement that. If there is a donor group present in the protein, then ligand should have acceptor. If there is a hydrophobic group, ligand should have hydrophobic group so that it can perfectly complement. Here you can see in the picture. See, this is the receptor. Blue color represents the hydrogen bond donor regions in the receptor, and you can see it is complemented by the ligand. So there is an acceptor group in the protein and there is a donor group from the ligand. Then it catches up a hydrogen bond. The dotted line represents the interaction. So here you can see the protein has a donor group and the ligand has acceptor group. And these yellow color regions are hydrophobic regions. So you can see all those yellow color regions are occupied by hydrophobic moieties in the molecule, right? So that is very much essential. So how do you find, I'll just give you a small imagination here, how a molecule is binding to the protein. Okay. Now imagine this is a situation inside the cell. The large molecule, what you are seeing is a protein. Okay. It's shown in a surface view. The ligand is inside the cell. Okay. You have taken through mouth. It has been absorbed. Now it is inside the cell. Okay. Inside the cell, you can see all the water molecules will be there everywhere. The ligand is floating and it is reaching to the protein. And while floating, did you observe it adjusted its shape and then it occupied its pocket in the protein, right? So this is just to give you an imagination. I'm showing you a graphical picture here. Okay. But how do you predict how a molecule can bind to the protein? And the process, the computational process, or the methodology we use in molecular modeling to predict the binding mode of molecule is called as molecular docking. I'm sure many of you might have heard it, right? Molecular docking. Docking means placing. You are placing a molecule inside the protein. Okay. So is it just like that keeping thing? No, you saw in the video, right? It's not just placing, it adjusts. So all those things we have to keep in mind, right? So there are two things when we are talking about molecular docking, two things we are trying to find out. The first thing is binding mode, with what shape the ligand is occupying the pocket. That is the first challenge. Okay. So now what is mean by that? Okay, let us go into the detail. The second thing is binding affinity, with what strength it is binding. Okay. So what is meant by binding mode? Let us try to understand. Okay. With what shape the ligand is going and occupying. That is what we are trying to find out. So whenever whatever objective you take, it always want to be in the lowest energy conformation, right? For us, lying down, if the bed is there, lying down is the lowest energy conformation, right? But you can't lie down whole day. Standing upright is a high energy conformation. You need a lot of energy to spend. You get back pain and all. So whole day standing is also not possible. So that's why you are provided with a chair or a desk in the classroom, right? Now, depending on the type of chair, your posture changes. So imagine yourself as a ligand and the chair as a protein. So if you are sitting on a tripod stool, your posture is different. The way you sit is different. If you have a plastic chair with back support, the way you sit is different, right? Then if you are provided with the sofa, the way you sit is different. What is the basic principle there? When you are sitting, you are comfortable and both you and chair should not fall down. The same thing applies here also when molecule binds to the protein. Okay, so it would try to take the best possible. Now, see, for us, lying down is the best posture. But when you are sitting on the chair, you don't lie down. You are not going to take lying down posture. But it is something different, right? The same thing also you should keep in mind. It's not the lowest energy conformation, but something different where both the protein and ligand would be comfortably sitting with each other. Okay. That is how the identifying that is called as binding mode. Okay. The second thing is binding affinity. So we already discussed affinities. They were driven based on the non-bonded interaction. So in the process, in the drug discovery process, we try to identify the molecule which can offer maximum number of interaction. You can see the molecule which is offering more number of interaction would exhibit higher binding affinity, and that is what we want to identify. So how do you find out? Now there are two challenges here: binding mode and binding affinity. So let us try to understand how do you find out that. First thing is binding mode, right? The comfortable posture for the molecule when it is occupying the protein. That is what we are trying to find out. So how do you do that? So any docking program you consider, okay, there are hundreds of docking programs available. Any docking program you consider, there would be two components in it. The first one is called as search algorithm. Okay, what does search algorithm does? So it generates different shapes for the ligand. What is meant by changing the shape of the ligand? If there is a single bond, okay, if you change the dihedral angle, this bond angle, if you change, then you can generate with different shapes. So like that, it will alter. Okay, if you rotate this dihedral angle, then you can generate with a different shape. You can see here, right? This is one shape, this is second shape, third shape, fourth shape. So that is what the search algorithm does. It generates ligands with different shape by rotating the single bond. Okay, dihedral angle of the single bond. Now there are various ways with which you can do this. Okay, there are different protocols. Random search protocol. Now this is a number, right? This is an angle. It will be between 0 to 360. So a random protocol program would generate a number between 0 to 360. So it could be first 10, then next it would add 50, then 60 degree it will become, then 60 plus another, let's say 20, 80, it will become 80 plus 100, 180 degree, right? So random number generator will be there. In random generation protocol, you have various sub protocols, something like genetic algorithm, Lamarckian algorithm, matrix method. You can check the details. I can't explain it here, right? So will be otherwise out of focus. Okay, but you can get the information from various sources. So that is one approach, random search protocol. Randomly you generate the number. But this, now the problem with random search protocol is it doesn't confirm exploration of whole of the conformational space. Something might get missed. So in Schrodinger, we use systematic search method. So a regular interval you add, let us imagine 1 degree. So every 1 degree you are going on adding. Okay. Now imagine, so there is one single bond here. So 360 shapes it has generated. So every one increment, it will generate one shape. So there is one more bond here, 360 to the power 2. Now there is one more here, 360 to the power 3. Imagine 360 into 360 into 3. All permutation combination we have to keep in mind, right? Then here there is one more, here there is one more. So imagine the number of shapes generated. Okay, this is if I give 1 degree increment. Imagine if I'm giving a increment of 0.1 degree. Now 3600 shapes it would be able to generate. And imagine how many permutation combinations possible. 3600 to the power 2 to the power 3, 4, 5, right? So like that. Okay. Now the second component is scoring function. So once it generates the shape, each and every shape would be taken and it would be put inside the protein, and it would analyze the energy contribution coming from each type of interaction, energy of van der Waals interaction, electrostatic, hydrogen bond, hydrophobic interaction, right? So from there, it will calculate the binding affinity. It's not the summation, it's some regression equation. From there, it would be calculating the overall binding affinity. Okay. So now it all depends on what type of the quality of the results, what you see for a software depends on what type of search algorithm you use and what type of scoring function you use. As I mentioned in Schrodinger, we are using systematic search method. That is why it is, there is no chance of missing out anything because you are exploring whole of the conformational space, and it's a smart approach. And the scoring function, what we use here is very unique in Schrodinger, so very accurate. So it considers all the energy contribution coming from all the type of interaction. It's a modified chem-score method we use here. So before you go into the docking, you need to process, you have to prepare the system. Okay. So for example, starting from the ligand. So when you sketch a ligand, you represent it like this. In any chemistry textbooks and all, you see 2D representation, but molecule doesn't exist in 2D, right? It exists in 3D. So the way molecule actually exists in the nature, the same thing you have to represent in the computer, then only you get meaningful result. So when I convert the structure, it's not just the geometry optimization, not just the conversion of molecule from 2D to 3D. I have to take care of many other things. For example, ionization state. Okay. So every atom, ionizable atom, we calculate the pK, and according to the pH what you're defining, we calculate what is the ionization state. Okay. So what is the tautomeric forms possible? What is the chirality definition? So all those things we take care. Okay. Now ionization state, it looks very simple, but keep in mind, even though it is very small thing, this will have a major implication on the quality of the results. See this particular nitrogen here, I have put a picture. So if it exists in the unionized form, you see it is exhibiting different type of interaction. When it gets protonated, you can see here the way it exhibited interaction is totally different. So now you have to be really accurate. You need to know whether it exists like this or whether it exists like this. Or if you don't know, at least software should be really good enough to say it would exist like this or it exists like this, right? So that is very important. That is where Schrodinger tools are very accurate and very very good quality results you can get from the tools. The way you prepare the small molecule, similarly you have to also prepare the protein structure. Okay, we will be discussing all the detail when we are having hands-on, so I'm very briefly going on here. Okay, so similarly you have to prepare the protein. So protein structure you can get from a database called as PDB, protein data bank. There are different techniques available: X-ray, electron microscopy, NMR structure. Okay, how do you select all those things we will be discussing later. So here also you have to prepare. So many of the time X-ray structure, no, never, unless it is ultra high resolution structure, X-ray structure would never have hydrogens. Many side chains might be missing, many residues might be missing. If the hydrogens are not there, then you don't know what would be the ionization state. And there is larger complexity in the protein because depending on the microenvironment, the way they exist may change. The ionization state may change. The same aspartic acid in one place, it may exist as neutral form. In another place, it may exist as ionized form. Histidine can exist in 5 different tautomeric form depending on the microenvironment. So in Schrodinger, we have a tool called as protein preparation wizard. In single click button, it would be able to prepare, it would be able to process all these issues. The next thing is you need to know where is the binding site of the molecule, where the molecule would go and bind. So if already you have taken a co-crystal ligand, so experimentally you already know where is the binding pocket, so you can explore the new molecules in the same site. If you are not aware, if you don't know where is the binding site, then in Schrodinger we have a very robust tool called a site map which can predict the binding pocket. So it will rank order, it will analyze the environment of the pocket, and it would suggest you give first preference to this pocket, second preference to this pocket, like that. It would tell you druggability score and also site score. Okay, for docking we have a tool called as Glide. Okay, so the advantage, greatest advantage with Glide docking tool is you have a choice between speed and accuracy. So there are three different modes available. HTVS, high throughput virtual screening. You you might have heard high throughput screening. That is a robotic arm will be there, very large number of molecule one by one you pick and then you experimentally analyze the activity. So here it is virtual. So that's why high throughput virtual screening. Just 1 second it takes. So to dock one ligand, it will just take 1 second. Okay. The next mode is standard precision, little bit higher accuracy mode it has. Okay. This will take 10 seconds to dock a molecule. Okay. One ligand docking will take 10 seconds. Okay. And the most accurate mode is called as extra precision mode. This will take 100 seconds to dock a molecule. So HTVS will take 1 second, standard precision will take 10 seconds, and extra precision mode will take 100 seconds. So what is the difference? Conformational analysis. In case of HTVS, the shapes generated for ligand would be few thousands. In case of standard precision, it would be lakhs together. In case of extra precision mode, the sampling is also more. Not only that, the scoring parameter is also different there. We will discuss when we go into the detail when we have hands-on session. So we use a grid-based approach here. We map the binding site information onto a grid. So what is the environment, what type of amino acid residues are there, what type of interaction they can offer, all those things we map onto a grid. Here is a comparison. So you can see the enrichment, that is ability to identify a true hit, okay, from a population is very high compared to any other software for glide docking, and even it is very efficient. It is highly accurate in predicting the experimental course. So with this, I'll stop. So this is my team. You already heard to Raghu. This is myself. This is my colleague, Dr. Sudarshan. He's a material science expert. Mr. Arun is our office admin. You already heard about her to Dr. Prajwal. Dr. Vinod and Dr. Kaushik are helping us here in the workshop. So you would be interacting with them soon. He's Kishore. He's the one who set up the cloud instances. And in some time, you will hear also to Shelvia. Shelvia is our account manager. If you are looking for purchase and all related to the software, then you can talk to her. So with this, I'll stop my presentation. If there is any question, I'll be happy to take it. Thank you.
Yes, thank you very much, Dr. Pritesh, for such a nice introduction on the topic. So it seems there are two questions given in the chat box. So first one is the inhibitor should have reversible binding. That's the question.
Yes. So traditional approach has been to have a reversible binder because we don't really want to permanently damage the protein. So that's why people prefer to have non-bonded interaction. They did not want to covalently or denature the protein. But nowadays, with the seriousness of the disease condition, people even have even are exploring the covalent inhibitors. So especially in the case of cancer and all, where the life-threatening disease is there, they really want to try out everything. So saving life is more important than other side effects. So that's why people have even explored. But one has to really keep in mind what is their objective here. So if the generally what happens when you permanently damage the protein, you get lot of side effects. So whether it is can you really compensate for those side effects? Can you save the life taking care? Like if you see when you start with the chemotherapy, the hair loss will be there in case of cancer treatment, right? So those type of side effects. So if the side effects you can nullify with its better effects, that is life-saving effect, then people would go even for irreversible binding.
Yeah, thanks. Second question was can preclinical studies be shifted to in silico?
Not really. Not really. Preclinical studies definitely you have to do, but the number of experiments you need to carry out you can really reduce. Let us imagine now in the earlier scenario, let us imagine there was no computational studies there. Okay, so let us I showed you the data, right? 2000 molecule they were synthesizing, all the 2000 molecule they were exploring. Nowadays you don't have to really do that. Through the computer, like we have technology, free energy perturbation and all, which are very accurate. within 1 kilocalorie, 0.8 kilocalorie of experimental result, now we are able to predict. The computational predictions are matching exactly with the experimental result. Still, you need to confirm that. So from 2000 molecule, you can reduce it to something like 10 molecules or 5 molecules. That type of scale reduction you can do, but definitely you have to try it. But the number of experiments, the extent of experiment you need to carry out, that can be reduced, and that would reduce the cost, time, and all. That is how it is advantageous.
Yeah, thanks. The next question is, what is the biological significance of having allosteric site in the enzyme?
It all depends on your requirement. So let's say now, let's say you are working on a kinase enzyme inhibitor. Now kinase is activated through substrate is ATP, right? There are 500 different types of kinases in the body which do different types of modulation, and all of these are modulated through ATP. Now, if you go for competitive inhibitor, that is substrate binding site, if you explore, there is a chance that the molecule what you have designed for one kinase could also bind to some other kinase, because binding site would be same, right? The same ATP is binding. Rather, if now I want to bring the selectivity, I don't want to disturb other proteins functionality. They are working fine, right? Only one protein is problematic. I want to control the activity of that. So if I target the allosteric site, that would be very unique for this one protein. So my designed molecule would only go and touch the unique protein, the single protein. So that is how strategically you have to think. So whether going with the, see, to control the disease, ultimately you have to prevent the conformation change. Now, whether you want to go with competitive inhibitor or allosteric inhibitor, that depends on the strategy and objective. What is the requirement? Based on that, you come up with the um, planning whether the allosteric site exploration would be good or competitive inhibition would be good.
Yeah, thanks. So the next question is, protein tends to change conformation through some external agent as well. So can we find out what shape would arise due to effect of the external agent through the search algorithm?
Yes, you can. Now see, depending on what is your objective, you have to explore different types of tools, different type of applications. Okay. Now I showed you in the glide, there are 3 different modes I showed you. HTVS was very fast. Standard precision was taking 10 seconds. Extra precision was taking. What I want to highlight here is, if you want to target complex thing, then you have to explore more complex type of tools. So when you are going with glide is a rigid protein flexible ligand methodology. It doesn't consider protein flexibility. But now you want to consider the flexibility of the protein as well as the external media, the ligand. Then you have to go for molecular dynamic simulation, where you make full system dynamic in nature. You don't put any restriction there, so it would be able to take any particular shape, and then you can monitor how the conformation of the protein is changing, or if you take any other external environment, any other solvent, other reagents, if you take how the conformation will change, all those things you would be able to monitor.
Yep. So next question is, why HTVS glide has less accuracy?
It's obvious, right? So if let us imagine there is a single bond, okay, one rotatable bond in the molecule, okay, one bond with which you can change the shape. Okay, now how can I put it? Okay, so this one bond, now if I go with the HTVS, as I mentioned, so let me share my screen again, back to, oops, sorry, 1 second. Okay, so here you can see, let us imagine this is the only bond I have. Okay, so now let us imagine with the case of HTVS, the number of conformers generated would be less. Let us imagine it is giving a 10-degree increment. So first angle would be this. Second shape generated would be with this much large angle. Then another one will be this much large. Another one will be this much large. Let us imagine when ligand is occupying in reality, when experimentally you do and when reality it is occupying, The angle is in between here something, but that search algorithm did not generate that. Now you missed out, right? So that is why it is less accurate. So if you generate all the confirmations, then you get more accurate result. So if you explore more confirmations, you get more accurate results. So that is what is done in standard precision, where the conformational analysis is more. Even in extra precision mode, the conformational sampling is more. Not only that, in extra precision mode, even the scoring function is further refined. We will discuss all the details when you are having hands-on session.
Yep. So the next question in the line is, nowadays organ on chips are coming in to build organ specific microenvironment at miniature level. So how efficient are these platforms for drug testing relevant to the time and the cost of drug discovery?
So many of the people are working on these type of technology development, and computational tools are really helping out because you can bring a rationale through which you can increase the efficiency, right? So immobilization of enzyme on the chip. So when now enzyme brings out some catalysis, so can it really efficiently bring that catalysis? So even in small concentration, can a molecule very efficiently bind? Can it be easily read by the sensor? All those things you can do. So to make better enzymes, so better sensors, people are working on modifying the protein, that is called as enzyme design technique. So a lot of people are using it, and people have been successful. Even many of people have been, you can even see a lot of publication coming out. Even people have been using it for even ingredient synthesis and all, green chemistry and all. So in all those areas, people have been using this type of technology.
Yep. So thank you very much. So I think that's the end of question sessions. So, okay, thank you very much, Dr. Pritesh, for such a nice introduction and answering all the questions patiently. And I hope all of the questions are answered from the participant side. If you still have any more questions, so you can unmute yourself and ask your question. Okay, so if there are no questions, then in that case, we can proceed further towards actually hands-on workshop. So for that, I'll just give you a brief information on how we are proceeding through this cloud-based workshop. So here you go. I hope the screen is visible.
Yes.
Okay, thank you very much for
Can you
confirmation.
hide your controls? The upper side control is there, no? That
Yeah.
is obstructing the screen.
Okay, so thank you very much again. And dear participants, so again, like welcome to your first experience on cloud-based hands-on training workshop. So first thing I would like to make clear is this is not just a webinar series on drug discovery. This is a hands-on workshop on Schrodinger software. So it requires your efforts also. So 50% effort we will put to train you, to demonstrate you the modules and various tasks, but 50% efforts should be from your side also. You also need to do the task, whatever are planned in this particular workshop. So this is what the idea of hands-on training workshop, and you have a cloud to do that. Software is already installed in the cloud, so you don't need to install anything in it. What you need is simply the web browser and internet, and most importantly, your time and efforts. So with this, I just give you brief introduction on how this workshop will proceed onwards. So first thing is the technical setup. What you need to have, you always need to join the workshop through laptop or through the computer. This is what very much important, because as I said, you also need to do the task, so you need to have computer with you. So it cannot be possible to do the task using mobiles or tablet, whatever you have. So you need to have laptop or computer. Second important thing is the Zoom app. So it is always encouraged to install Zoom app and join through the Zoom app, because you will have a lot of functionalities available through the Zoom app. So it will smoothen your experience. And the cloud login details for this particular workshop is already sent to you today morning around 6 or 7 o'clock. So just check your registered email ID and you will find the cloud login details, and those are your personalized cloud login details. So for your support, so we have a team of scientists who will be there to support you in all aspects. So you will able to interact with all of us one by one. So we are always there to help you out. And on the agenda, we will have few things which are planned for today's workshop, and it's specifically on Maestro graphical user interface. So Maestro is the graphical user interface for complete Schrodinger suite, which you are going to see it soon. And we also planned for the structure-based drug designing as a molecular docking example. So at the end of this workshop, you will be a little bit thorough through the molecular docking exercise and how one could use it for the virtual screening process and designing good molecules against the protein target. And then in next step, like the support structure which we have planned for. So how this workshop will proceed. So you will always have talks, so demonstration from our side. So we will give brief introduction on the topic, we will tell you how to do the task, and afterwards it's a hands-on time which is your time basically. So you need to do that task in your cloud and your software. So this is what the thing is, and for doing hands-on training we will also share with you the handouts. So those handouts will help you to do that task or understand the steps in pretty nice way. So next thing is, in between, to make the session interactive, we will have questions and polls, and we will have short discussions on it, so that it makes you more interactive, as well as makes us understand better whether you are getting the points or whether you need more information on it, and you will, at the end, like, you get more information. And you all participants will be divided into small subgroups, and this is called as breakouts. So for example, if you are 100 participants, you will be divided into 5 different group of 20 each, so that we will have better connection with you. So in each of the breakout room, you will have one scientist present to help you out with that particular task. And in this way, this breakout sessions will be proceed. So I'll tell you in more detail how it is. So first thing is regarding Zoom app. So everybody is kind of familiar with Zoom app nowadays. So the only thing which I would like to mention here is you can communicate with us by unmuting yourself. If you have any question, you can type the questions in chat box, or you can also share your screen if you are facing some issue and we need more information of it. So simply you can share your screen, and then we will see what is the issue which you are facing. So then time to time, whenever we have demo and hands-on going on, you will have such a pop-up window in front of you which asks you to join the breakout rooms. So as I said, you will be divided into small groups. This is called as breakout rooms. So you just need to click on the join button, so that you join that breakout room where you will have a separate scientist present there to help you out with the task. Then coming towards the handouts, so handouts are nothing but the PDF document which we are going to share with you for each task throughout the workshop. And it is just step-by-step guide on how to do the task correctly. So it has one-liners, it has images, it has markers. So what you need to simply do when we ask you to do the task, just go to the handouts, look from the page 1, step 1, and try to do the same way what is mentioned in the handouts. Try to read those one liners because these are important information giving you, and try to look where to click, where are the highlighters given there. So please do not skip any of the step from the handouts, otherwise you will really miss the step and you will not able to do the task. It's just a simple 5 to 10 pages of documents with images and markers. So it will help you to do that task correctly, and it's just a small task which you need to do. So most important thing is, please do not deviate yourself from the handouts. Try to follow the handouts exactly. Try to read it, and you will be able to do the task correctly. Then there might be various technical issues which you come across, and that's perfectly fine. So first thing is how to get rid of these technical issues is, if you follow handouts correctly, then 90% of time you will not come across with any issue. So handout is the ultimate thing which helps you out without without getting any technical issues. But even if you get stuck, you feel like, okay, I could not do the thing, please come back to us by chatting, by unmuting yourself, or sharing your screen. and we will be there to help you out. So this is what we promise you. The next important thing is there are certain instances when we can't really help you out, and those are like if your internet is really unstable or it is not working. So because this is internet based event, so internet connection must be there, should be continuously working, should not be slow. So this is what we request all of you, to make sure your internet connection is regular. And second thing is from your side, like you should not be irregular in joining or joining late the sessions. Otherwise, you will really miss a lot of crucial information given by the speaker. So please be with us. Please be regular and efficient in doing all the tasks and following the instructions. But at any given point, if you feel okay, I need to discuss it with scientists, I am facing some issue, please do not hesitate to reach out to us. We are there to help you out. Then next thing is the cloud infrastructure which we have. So we have deployed good cloud infrastructure from Google Cloud. So it's a huge cloud infrastructure which we have deployed for you for your smooth experience. So there are many GPUs available, there are many CPUs available in the cloud along with the software installed in it. And it has interface just like your desktop, but it is in the browser. So you will have a chance to see like how it is, and software is already installed there that you can directly use it for your task purpose. So there are huge computational infrastructure deployed for all of you, so no need to worry about the things whether it will become slow or not. It all depends on your internet connection, and everything will be fine. Then regarding cloud login details, so basically for login into cloud, you have a specific link which is sent on your email. So you must have got email at around 7 o'clock today morning on your registered email. Just check it, which has a mention about the cloud link. It starts with HTTPS NITR workshop, like that. And what you need to do, you just need to click on this particular link. It will direct you to the web browser, and then which shows you the virtual workstation. And in that virtual workstation, you need to put the username and password given for you. So let me be clear, this is a personalized username and password. It is only for you. Please do not share with anybody. And your username starts with user hyphen some number, and then password is Schrodinger hyphen some random number. So this is what is personalized. Please do not share with it anybody. And next thing is, this is case sensitive thing. So please do type it correctly or copy paste it correctly. So there is no error until unless you do it incorrectly. So please check your cloud login details on your registered email and log in through the cloud. So once you click on this particular link, which is given in your email, it will open the web browser and it will open your virtual desktop like this, in which you just need to put your user ID and your password correctly, case sensitive manner, and hit the login button. Once you hit the login button, it will open the another page, and it shows you two options like terminal for user and virtual workstation. So the option second is of our interest. What you need to simply, you just need to simply click on virtual workstation for user. And once you click on virtual workstation for user, you will see the dark color screen, and you will see the message appearing on your desktop is your desktop is being prepared. This takes a moment, please be patient. So just request is please be patient, don't worry, it will take some time. Sometime it may take 1 minute, sometime it may take 5 minutes or 10 minutes to prepare the system depending on your internet speed and how much the traffic is. So don't worry about it, it will get prepared, and once it gets prepared, you will see the virtual desktop looking like this in your web browser. And once you see, it is just like a simple your laptop or your desktop, just like what you are using in your regular things. So it has software installed in it, it has every option same as that of whatever is mentioned. So out of all those icons, you need to look for this M icon, which is a blue color one. So this is M icon is for Maestro. So once your desktop is prepared in front of you, you just need to double click on this Maestro icon. And once you double click on this Maestro icon, it will open the Maestro interface for you. And this is what the Maestro interface looks like, which is a gateway for Schrodinger software. And then in next task, we will see like how to proceed through it. So that's the only simple task which you need to do. You just need to click on the link, cloud link, log in into cloud and click on the virtual workstation option, and it will prepare your desktop, and click on the Maestro icon and it will open the Maestro for you. That's the simple 6 steps which you need to do at this point. So for that, we will have small breakout session. But before that, I'll tell you some few of the important points which you always need to understand. Is so whenever the speaker from Schrodinger is giving you the talk or the demonstration on particular task, please do not try to do the same task at the same point, because if you do the same task at same point, you will miss really a lot of information. So but we are giving you the separate time slot. After the demonstration, you will be given 15 to 20 minutes to do that task with the help of handouts in the breakout session in front of scientists. So you will have enough time afterwards to do that task. So please do not worry about doing the task at same time when demonstration is going on. While demo is going on, please focus only on the demonstration and try to understand the things whatever is going on. And other thing is important, please do not deviate yourself from by using mobile or doing anything else, because lot of information given by speaker and you will miss that information, then you will not able to do the task correctly. Please try to avoid the use of mobile or anything else as much as possible. That's the only request. Other thing is, even the workshop will last till 4 p.m. But even after that, you will have enough time to do the task in the cloud. So cloud instances will be on, handouts will be with you, and you will have enough time to do the separate tasks. If you want to use cloud for learning purpose, you can definitely use it. It will be available till 8 o'clock at the night. So don't rush into the task. Just for this particular workshop, try to stick to whatever speaker is demonstrating you. Other thing is, please do not run your personal project. Maybe out of you, few of you are proficient in using software. So please do not use your personal projects on the cloud, because it is not possible to download or upload the data. So don't run any job on it from your personal project. But whatever is for learning purpose, you can definitely use it, sticking to the input files which we have provided. You, as I said, you cannot download or upload the data in the cloud, and it has auto kill option, so it will not run many long jobs. So do not run your personal projects on it. And at the last, like, you can always contact us in case of any scientific queries which you have. We are always happy to be in touch with you, so please do not hesitate to reach out to us. With this, thank you to all of you. In case if you have any questions, uh, I'm happy to take it. Just post it in the chat box. So there is a question about, I haven't received my cloud login details. So just check your registered email ID. If it is correct, then you must have received it. I did not get any email bounce. So if you are not registered, then we don't have that facility to understand what is going on. So if you still want to use it, please share your email ID, so that I will check it and I'll send you the separately cloud login details. Then can we use cloud-based work at daily? So I'm sorry, that option is not available. This is only for cloud-based workshop which we have. So the instances will be on till 8 p.m. at the night. So you can use it for learning purpose, not for daily. Then is there any timeout of virtual machine? No, there is no timeout until workshop is going on. It will be always on. Then uh, so there are questions about handouts. So I haven't sent any handouts yet. I will share it with you in the Zoom chat box, so please wait for it. And in any case, if somebody is not yet registered and they want to use it, just send your email ID in the chat box, personal chat box, and I will share with you the handouts if it is possible. Then yep. So this is my register ID. Still did not get any email. So I'll check it out. Please be online and hold it. And so for all other participants, I'm just going to share with you the handouts for this particular task, which you need to do at this point, which is login into cloud and launching the Maestro. So here you go. Please check the Zoom chat box. Yeah. So whoever is not registered, don't worry. Just send your email ID and I'll check it with it. Okay. Yeah. Please do not share your email IDs multiple times because once you paste it, I got it. Okay. So that's it. So I have already shared the handout links in the Zoom chat box. Just please check the Zoom chat box. It is about how to log in into cloud. It is a Google Drive link for handouts, simple PDF document. Just click on it, see the document and try to follow it exactly as mentioned in the document, and log in into cloud and launch the Maestro. This is the only simple task which you need to do it now. And for that, I'm creating a small breakout rooms. So, Kaushik, could you please confirm how many breakout rooms should I create?
I'm not sure if Sylvia is here. If she is there, we can have 4. Otherwise, we'll have
Four.
3.
So, Shelvia, are you online?
Hi Prajwal, sorry, I'm on a call.
Okay, sure.
Okay, let's just do
No
3
problem.
then.
Okay, then I'll just assign 3. So here you go, all participants. So breakout rooms are open in front of you. Please do join the breakout rooms. And try to do the task as mentioned in the handouts. And in case of any queries, please reach out to the scientists present in your breakout room. Thank you. Yes, welcome back. So thank you very much for joining. And for most of you who already pasted your email ID and requested for the login details, we'll try to send you as soon as possible. It may take some time, but don't worry. In meantime, focus on the next demonstration, and I'll try to send you soon. So please be patient and focus on the demonstration. And now we can proceed for the next demo. For that, I would like to request my colleague, Dr. Kaushik, to help us with the demonstration. Thank you.
Thank you very much, Prajwal. I hope you can see my screen. So yeah, as was mentioned by Prajwal, for those who haven't received the login details, don't panic. What I'm going to show right now is quite basic, so you're not going to miss much. And we are just going to start build up slowly into the hands-on activities of the workshop. So I hope all of you can see my screen. If you don't, just let me know. So all of you should be looking at a screen that looks like this once you have logged in. So you just have to click on the icon that you see Maestro here, which will be the application that we'll be using for all the activities that we're going to do today. So I'm going to launch the Maestro here. Initially, you're going to see this terminal pop-up, and you're also going to see another application pop-up later on, which we'll be using for all the calculations today till the evening. So this is how the Maestro looks like, and your software might initially be to the top left of your screen. For a better experience, just maximize the window. So it's all basic stuff that people would usually do. So now I have this whole screen is fully occupied with Maestro software. And so there are different aspects to your docking. So first thing is you're going to worry about the ligand molecules that you want to dock into a protein and see how they interact with the protein. So there are two components. One is the ligand molecule, the drug molecule, and the second component is a protein molecule. So we'll take a look at how to interact with those ligand molecules or the protein molecules step by step in this in the next few demo sessions. So this is the Maestro interface here, and there are quite a few buttons that you can see. There are quite a few options you have here, and you also have some options here, and you have a few options here, here. All of these things are there. So instead of trying to show you all of them at the same time, right now, what I'll do is I'll slowly build up the whole demo session where we're going to take a look at each button one by one and how it works, depending on the task you want to perform. The main thing that you have to be aware of is that in this Maestro interface, you have this black area, which is called the workspace. So in the workspace, any molecule that you load into the software will be displayed. So this is a place where all the molecules are displayed, and you have the workspace navigator where you can find out all the molecules that are part of your project. Some of them will be shown here. Some of them will be displayed, will be just like the list will be there. You can just choose which one you want to show in the workspace as we go along. So as I mentioned here, we're going to be dealing with drug molecules. We're going to be dealing with protein molecules. So let us try to sketch a simple drug molecule and see how you can add drug molecules to the software. So before you do those things, there are two things that you have to do anytime you launch Maestro. So the first thing that you have to do is you have to go to the file option here, and you have to go and change your working directory. So the reason why you have to do this is that when you're going to run the calculations, you're going to have lots of files being generated. And if you don't save your working directory, then the files will be saved somewhere by default, which is not the place where you want to be. So you have to keep searching for where the files are, and it will be complicated. So to make things easier, you can just like change the working directory and put it in a place that you know exactly where it is. And all the files that we're going to generate today during the calculations will be saved in that working directory. So right now I'm in my home directory. So I'm using user62, and you all will have your own usernames. So I'm going to go to a desktop folder here. And in the desktop folder, I'm going to create a new folder for today's workshop. So I'll just click on this create folder option here, and I can give a name for it. And since we're going to work on docking today, I'll just call this as docking calculation and press enter. And that will be my working directory. So once I have created my working directory, the next step is to save your project. Many of you would be playing video games right now, and you know that in video games, you have like autosave options where it saves your particular game at a particular time point, so that you don't have to start from scratch every time. You can just continue from where you left off. In the same way for the software also, if you save your project once, it's like saving your game session, then it automatically saves your project at different time points, and then you can just continue from where you left off. If you have a power cut or if you have some other issue by which you lost your project details, you can always like go and open your project and you'll have everything the way it was before. So you can go to file and click on save project as, and you can go, it will automatically point you to your working directory that I have just created, which is home user 62 desktop docking. And I'm going to give a project name here. I'll call this also as docking, and I'll click on save, and that will save my working directory and also my project. So with these two established, the next thing that we are going to do is now we'll start adding molecules to our Maestro software. We'll take things step by step, nothing complicated here. So to add a molecule to your Maestro software, there are multiple different ways of doing it. There are 4 to 5 different ways of doing it. I'll show you one of the ways right now, which is something called the 2D sketcher. So you go to edit here, and then you go to something called the 2D sketcher, which will allow you to sketch a molecule in this particular instance. So this is a 2D sketcher, and you can also observe that on the left side of this 2D sketcher, I have quite a few basic structures available for me and a few basic elements available for me, like carbon, nitrogen, oxygen, all of these things. And you also have options like draw bonds, all of these things. We'll take a look at each one by one. And so for this particular demonstration, I want to show you how to sketch a molecule that most of you must have taken in your lifetime right now, which is paracetamol. It is something that people are often prescribed when they have fever. And so let's take a look at how to sketch a simple paracetamol molecule. So to show you how the structure of paracetamol looks like, I can just go to the browser here, which is my Firefox browser in the cloud instance. And once a browser launches, I'll show you how that molecule looks like. And so I'll go to the Wikipedia entry here, which will have the structure for us, like a brief image of it. So this is your paracetamol molecule. So if you can take a look at it, you have an aromatic ring, then you have NH group, you have a acyl bond, you have a methyl group at the end, and you have a hydroxyl on the other side of the benzene ring. So if you want to add this, you can see that the basic thing that you need is the benzene. So if you just go back to your software here, you can see that here I have a cyclopropane, cyclobutane, cyclopentane, hexane, and you also have like heptane, octane, and you have cyclopentadiene, and you have the benzene. So you just click on the benzene here in this list. And once you click on the benzene here and put your cursor anywhere in the white area, you can just click anywhere, and that will place your benzene ring in that particular position. So once I have done this, the next thing is, if you observe, remember the structure, I need to have like bonds 1, 2, 3 here. And I also need to have 1 here, and also need to have 2 bonds here. So to add the bonds, you have this draw option here, which you can choose from this option here, the 4th option on the first column. And you just click on one of the atoms of this benzene ring. So for example, if you put a cursor here, you can see there's a gray circle that forms in the carbon atom. And you can just click on that particular atom, and it's going to add a bond to that particular atom over there. So it adds a carbon by default. And we have to have nitrogen and oxygen here, but we're going to worry about that later. For now, I just want to like add the bonds to make it like longer. So I'll just like add 1 more here, and I'll add 1 more here. So this particular arm is like, so far I got 1, 2, and 3. Now I need to add like 2 more bonds here. So for that, I can click on this carbon again. And because it already added 1 group here, it's going to add the other one in this direction here. So I'll just like click on this particular carbon again, and you can see that right now it added a bond in this direction here. And this has to be a double bond, not a single bond. So to make it a double bond, before we're clicking on each of the atoms here to draw a bond. But if you want to make a single bond to a double bond, you just click on the bond here, and that will make it into a double bond. And all of these things are mentioned in the handout, so don't try to memorize what I'm showing you, and also don't try to do it at the same time as I'm doing it. We'll give you enough time to practice these things in the breakout sessions, so don't worry about it. And the handout has all the instructions for you to go through one by one. So all I have done is I have the single bonds right now. I just want to make this in a double bond here. So I'll click on the bond itself, and that will make it a double bond for me. So this particular arm looks okay. Of course, I still need to make this to nitrogen. I still need to make this to oxygen here, but I also need to draw one more bond in this direction here, which will be my hydroxyl group on this particular molecule. So to make it a hydroxyl, I can just click on this particular atom here, which is opposite to this particular arm here. And you can see it has drawn another methyl group for me, for this benzene. So now that I have done this, now I need to start replacing this carbon with an oxygen, this carbon with an oxygen. So to do that, you can just click on the oxygen atom here, and you click on this particular carbon here, and notice that right now it has converted the carbon to an oxygen. It also added hydrogen to the oxygen automatically to satisfy the valency of that oxygen. Oxygen has a valency of 2. One of the bonds is interacting here. The other one has to satisfy with the hydrogen itself. That's why it adds hydrogen automatically to the oxygen. If you do the same thing for this carbon here, now since you have double bond here, this oxygen will not have hydrogen to it because the balance is already satisfied with it. Now, finally, for this paracetamol molecule, I had to convert this carbon to a nitrogen. So you click on the N option here, and I click on this particular atom here, and that will convert it to NH automatically because nitrogen has valency of 3. You have one bond here, and a third bond is with the hydrogen here. So this is our paracetamol molecule that you have simply sketched with a few simple clicks. So all I have done is in this particular demo session so far, I went to File, change working directory, save the project, and then after that I went to Edit. I went to 2D Sketcher, and in the 2D Sketcher, I clicked on the benzene ring, clicked in the white area. Once I have the benzene, I click on the draw option here, click on this atom here to expand this particular group here. And then after that, I clicked on the oxygen to make sure I can convert the carbon to oxygens here. I clicked on the nitrogen to convert the carbon to a nitrogen here. Now, this is what we have sketched so far, and the molecule is still not yet in my workspace. To bring it into the workspace, I have to click on Save as New option here, and it will ask you for the name of the molecule that you're trying to add. So since this is paracetamol, I'll just call it as paracetamol, and I sketch this in 2D, so I'll give a name as _2D here. And I'll click on OK, and notice what happens when I click on OK. Right. Now you'll notice that I have the molecule in my maestro here, and the name of the molecule is shown here. And you can take a look at this. You have your benzene ring, you have your hydroxyl, you have your amine group, you have a carbonyl group, you have your methyl group. So you have simply, with a few simple steps, you have sketched your paracetamol molecule. So it's as easy as that. Prajwal, do you think we should take a break right now, or should I go for the 3D builder also and then go for a break?
So I think we should take a breakout. Okay. And just to add to that, whoever requested for the cloud login details, I have already sent email to you. Just check the registered email. Please do not paste your email ID again and again. Once it is recorded, it is recorded. So don't worry about it. Just check the email ID and you will see the cloud login details, and that you can use it for your login and doing the task. So to do the same task as demonstrated by Dr. Kaushik, just check the Zoom chat box. Please do not flood the Zoom chat box with lot of email IDs. So just check the Zoom chat box. I have shared with you the link for the same demonstration. So please go to the handout link. Try to follow the small task of drawing 2D structure exactly as mentioned by Dr. Kaushik as well as in the handout. And then let us know if you face any difficulty. And for the same, I'm just creating breakout rooms. So please do join the breakout rooms and try to interact with the scientists present in your room if you face any difficulty. Please try to do the task and let us know once it is done. So here you go. The breakout rooms are open for all of you. Please do join the breakout rooms. Thank you. So welcome back. I hope you are able to do the 2D sketcher and converting the structure into 3D. So now we can proceed for the next step. Over to you, Dr. Kaushik.
Sorry. So we have built this molecule in 2D sketcher. And now we are going to explore, as mentioned, there are multiple tools using which you can try to load molecules into Maestro. We will explore a second tool right now, which is something called the build option. So you can see here in the Maestro that you have the hammer and the spanner. It's just like a mechanic builds things. We also have like tools for building your molecule and repairing them or fixing them. So to use a build tool, you just click on the option here, which is in the center of Maestro here. And you have something called 3D Builder that opens up. So 3D Builder is a tool using which you can build the molecule directly in the workspace. You're not going to do it in 2D Sketch and import it. You're going to build it directly in the workspace. So what I'm going to do is I'm going to click on this first icon here with the plus sign, which will create a new blank entry that is part of my workspace. So right now it's called structure 2 here. And there is no molecule yet. We're going to add some fragments to it later on. But for now, I want to sketch the same molecule, which is paracetamol. And I'll do it in 3D directly instead of trying to do it in 2D. So now that I have given a name for the molecule, I'll have to add fragments to the molecule. Just hold on. I want to clear the drawings that people have done. Okay, great. That's taken care of. Great. So to add fragments to this molecule, which there is no fragments right now, you click on the add fragment option here. And that will show you the similar options that we have explored before. You have your cyclopropane, pentadiene, benzene, all of these things. You have your hydroxyl, amine, sulfone, carbonyl, methyl, all of these things here. And so if you want to sketch paracetamol, just as we did before, you take your benzene ring. So in the previous time, we had to sketch the whole molecule before we could add it to the workspace. But right now, if I click on the benzene ring, it will add the benzene automatically to my instance here. So just click on benzene here and you notice that the benzene is automatically in my workspace. Now, for this benzene to sketch the paracetamol, again, I need to make sure that I can grow the molecule in this direction here. I can also like add this hydroxyl here. So I'll start with the simplest ones. I want to add the hydroxyl for this particular molecule. So to do that, what I'm going to do is I'm going to click on this particular hydrogen here. You have this benzene here, you have carbon, 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 carbon. And these white lines that you see at the edge of each carbon is the hydrogen atom that is connected to your carbon. So benzene has C6H6 is your molecular formula for benzene. So this is one of the hydrogens. I'm going to just choose this one here. And I'll click on this particular atom here. And now you can see that there's a blue circle that appears on this particular atom. So this means that this atom is selected and I can try to replace this atom with anything of my choice. So I have sketched a benzene so far, and then I have selected this hydrogen atom here, which I'm going to replace with a hydroxyl right now. So selecting this, making sure the blue circle is selected here for this atom, I will click on the hydroxyl for this particular panel here. And you'll have the hydroxyl added to your benzene ring automatically. So now that I have done this on this side, on the other side, I'm going to select this particular hydrogen here. And this hydrogen, instead of like, previously in 2D sketch, we made it like, bond, 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 then we try to replace it with nitrogen and oxygen, all of those things. But right now, what I will do is, instead of trying to do that way, I can directly add amine group to this particular region here. So I selected this hydrogen atom here. I click on this amine group here, and that will add my amine to this region of my molecule. Okay, now I have an amine group, but now I need to convert this amine and I need to grow the amine to form this particular bond here. So to grow it further, I can select the hydrogen that is part of this amine group here. So now I select this hydrogen here. And once I have done that, I'm going to add a methyl group, because right now you have a methyl first and one more methyl, and then after you have this one here. So for that, So for that, I'm going to click on this methyl group here, and that will add my methyl here. And I also need to have a methyl here also, which I need to extend, which I'm going to add one more methyl here. So now I have this. So far for this molecule, I got this hydroxyl, I got this benzene, I got this amine group, I got this particular carbon here, I also got this carbon here. But I need to convert this to a oxygen. So I have this particular carbon, which has 2 hydrogens right now. I need to convert these 2 hydrogens to an oxygen and make it into a double bond here. So to do that, if you observe what I have done so far, every time I want to change something, I just click on the hydrogen atom that I want to modify and click on the atom of my choice. So in the same way, if I want to convert this hydrogen to an oxygen, I'll just click on this hydrogen here and make sure I have blue circle on it. And I'll click on the hydroxyl here to make it a hydroxyl like this. Now, this is not complete yet because I have a OH group here and I need to have a double bond O, and I still have one more hydrogen here. So I need to convert this particular C single bond oxygen to a double bond. And for that, what I will do is instead of clicking on the atom here, I'll click on the center of the bond here. So I have this particular bond here with the COH bond here. I just click on the center of this bond where you can see this gray line intersecting with the red line here. Red is for oxygen here. If you click on the center, it will automatically select the 2 atoms that are part of your bond. So I just clicked on the center of where you have the gray and red lines intersecting here. So once I have this selected here, I can increase the bond order for this particular bond from a single bond to double bond by clicking on this option here, where you have the increased bond order option here. And once I do that, notice what happens here. So I did that, automatically the hydroxyl got converted to a simple oxygen, and the other hydrogen that was part of this carbon got deleted, and now you have a complete molecule which has the appropriate valency for all your atoms. So this is how I built a molecule in 3D here by just choosing a few options here. And before I can go for like any calculation, I have to make sure that this molecule is minimized. So I just drag my cursor around all of these atoms to select all the atoms here. And I click on this particular arrow here, which will try to minimize the molecule. Notice what happens, like this is a quite already stable molecule. So you have very little scope for improvement. It will most likely modify this group here, which can change its space where it's located at. So just when I click on this minimize atoms here, you notice that this particular part of the ligand will change. It will adjust itself and give you a minimized structure, which has appropriate bond lengths and angles for your molecule. So just to point out, the power of 3D Builder is not just about the characteristics of simple molecules here. If you click on the 3 dots here, in this particular panel, so I'm just going to show these 3 dots here. You have lots of pre-built fragments already present in this 3D Builder. So if you click on the drop down here, you have organic fragments. You can choose like any of these organic fragments here. That will add it to your molecule. You can go to like heterocycles here. You have so many heterocycles available for you. You have like carbohydrates available, which you can add simply by clicking on glucose, galactose, or whatever you want to add to this space. You can add like amino acids also if you want to add those things. You also have non-standard amino acids. You have lots of diverse fragments here. You can even sketch DNA and all of those things. In the diverse fragments, you have a whole list of fragments, methyl, ethyl, propyl. So you don't have to do one by one all of these things. If you know what group you want to add, what fragment you want to add, you can just click on that fragment and it will add it to your molecule automatically in this 3D Builder. This is mentioned in the handout, but we don't have time right now. But if you have any questions, you can reach out to us in the breakout rooms. Yeah, please.
Sir, I have a question, sir. For metal centers or metal type of drug, just as cisplatin, oxaplantin, sir. Is this able to working on that, sir, in metal centers?
Yeah, you can do all of those things. Well, there are dedicated builders for metal builders also. But for example, in the 3D Builder, right, you have an advantage of going to like this carbon here, and you have the basic elements like carbon, hydrogen, oxygen, all of these things. But if you click on the drop down here, just hold on, like, yeah, please go ahead.
Sir, previously when I used this Schrodinger software, sir, when you're working with the metal or higher dry element, Zinc 2+ or any type of metalloids in our lab in NITR, under Prabhavartam, this is not able to do it, sir. That's it. Is there any new type of algorithm or system is available now?
Yes, yes. So, well, one thing I want to point out is that you have a whole periodic table using which you can try to build all the elements that you want to have. But for your particular case, I would probably go to like, so this is not part of the workshop, but I just want to like quickly show you. So if you go to task option here, which I'm going to come to later, you have something called the materials. And in the materials, you have structure builders, where you have tools dedicated for building metal complexes, single complex, multi complex, all of these things. You can even build nanostructures, nanoparticles, all of these things also here. So all of these are present right now, which makes it very easy to build a complex. You can choose your single complex here. Again, this is not part of it. You can choose your central atom, which is a metal coordinating atom. You can choose a geometry for it. You can add different components here, and you can just build a molecule with very few simple steps. Okay.
Okay, so
So
this
for
is
example, sorry?
Sir, this is now a new addition to this software.
It's been there for like 2 years now. So I don't know how, when you did the previous calculations, but it's been there for 1-2 years easily right now. Right. So for example, I'm just trying to deviate here, but this is a molecule that I built, which has a selenium here. And I just like added these groups here, or maybe this one is more clearer for you. And I could build this molecule in like less than a minute with the prototype bond geometries. Okay, that's the selenium in the center over there. So again, that's not part of the workshop here, but I just want to show you briefly. We have all the tools available for building any molecule of your choice. As I mentioned, there are so many different ways of adding molecules to your software, to Maestro. And so this is one place where you can build like metal complexes, nanoparticles, nanotubes, nanosheets also. You can even build like disorder systems with different co-solvers, all of those things. Okay. So I hope this demo was clear. And for metal complex, I would actually recommend you to go through these options here instead of trying to build from the build option here. So this build option is mostly for like organic molecules. You have metal centers here, which you can add and try to like go through the metal centers here and metal ligands also here. But again, it all, I would rather start with the structure builder tools instead of going through these ones here. But this 3D builder is like quite handy when you want to sketch some molecule. Just to show you a simple thing, if I want to add tertiary butyl here, I can just click on this tertiary butyl here, and that will add it for me. Like it's as simple as it. You don't have to sketch everything one by one. So this is what I wanted to show you right now. The handout also show you how you can sketch like DNA molecules and things like that, but you don't have the time for it right now. So if you have any questions on other things, we can address in the breakout room. But for now, it will be nice if all of you can go through the 3D builder and see how you can build those molecules. Can we use miles of molecules as copy paste here? Yes, we'll address it in the next demo session, which is what I'm going to take a look at exactly next demo session. Thank you.
Yep. Thank you very much, Dr. Kaushik. So, dear participant, just check the Zoom chat box. I have shared with you the link for handouts. So it's the same handout which you used before, but now your task is to follow it from page 13 till the last of handout. So please do follow the handouts and let us know if you have any queries. For that, I just opened the breakout rooms. Please do join the breakout room and try to interact with the scientist while doing the task. Thank you. Welcome back, all of you. And I hope you are able to build the molecule using 3D as mentioned in the demonstration. So now we can proceed for the next step towards using Maestro. Thank you.
So far we have looked at like two different ways of trying to add molecules, and there was a question: can you add smile strings also into Maestro? So yes, you can do that. I'm going to take a look at it right now. So before you do that, I'm going to clear my workspace. I'll go to workspace here, click on clear workspace. So the molecule is still part of my project, but it's not shown in the workspace. So how do you add a smile string to Maestro? So you can just go to paracetamol again in the browser. So inside a cloud instance, not outside. And here on the right side, if you go down the list here, you have the smile string available for you for this molecule. So you can see the smiles here. Click on the show option. And this is a smile string, which is a representation of what your structures looks like in terms of the bonding and everything, connectivity. Select that whole string here, press Ctrl+C to copy the string, go back to Maestro and press Ctrl+V. Right? That's all you have to do. So it's as easy as that. So I just like not this one. In the cloud instance, I went to the Wikipedia instance itself, the same thing. I just went to like show here, copy this string here, went back to Maestro. Sorry, not this one. Where is it? Oh yeah, here itself. And I just press Ctrl+V here in the workspace, and I have a paracetamol molecule. And you can see that there's a new molecule here, which has a smile string for this. Just hold on. Yeah, just hold on. Yeah, one more minute. Yeah, sorry about that. So I just showed you like this one here, how to add a molecule as smile strings. And now you can see the smile string is the name of the molecule here. So if you want to change the name here, you can just double click on this entry here and you can type whatever the name of your choice. I'm going to like call it as paracetamol _ smiles. So now we have all of these molecules built. So we have like, so far we looked at three different ways: a 2D sketcher, a 3D builder, and a smile string. Of course, there are other ways also. So for example, I showed you the structure builder options over there. And you also have ways of importing molecules. And what we're going to do next is, in the interest of time, we have like 10 minutes left before the lunch break. So I'll start discussing on the protein visualization part. So we have so far seen how to import your ligand molecules. Now we're going to take a look at how to import your protein molecules into Maestro. So for importing your protein molecules, you can go to File here, and you can go to Get PDB. So PDB is a repository of protein molecules. It's a protein data bank. That's what it stands for. So you just like open the Get PDB here, and for this particular for today's activities, you're going to use a protein with the PDB ID. I think it is 6B2Q. And so I'm now going to download chain B for now. And in the post-lunch session, we're going to deal with chain A. But for now, to keep it simple, I'm going to load chain B just for demonstration purposes. So once you enter your PDB ID, like you don't have to go to PDB and like search for the molecule and download it. You can directly get it from here. You can just type your PDB ID here if you know what protein you're working on. Give the chain name here and click on download, and that will load your protein molecule automatically into Maestro. That we will see how you can visualize the protein molecule. Right. So there you go. We have loaded this protein molecule. And so this is, right now it looks like a big mess to me. I have seen so many different lines here. Some of them are in blue color, some of them are in gray color, some of them red color, some of them in green color. I have no idea what is what. And so I want to make sure that my molecule is better visualized. So far I have shown you the 2D sketcher option, I've shown you the build option, and now we're going to take a look at a few other options that will help us in trying to change the visualization of your molecule. So one such option is that for this one, proteins have so many atoms, so it's very difficult for it to get a feel for what is happening, how the protein structure looks like. But what we know is that we know that proteins have alpha helices and beta strands, which makes it a bit more easier to break down the structure into different components. So if you want to take a look at where the alpha helices and beta strands are in this protein, you can just go to the style option here. And you just click on the ribbon option here. And this will create a cartoon representation for your molecule where you can see your alpha helices and beta strands automatically. So all I have done is I just click on the style option and click on the ribbon option here, which again mentioned the handout. And you can see that with a couple of clicks, I got all of these alpha helices and beta strands. The coiled ones are alpha helices, and the ones with the arrows are your beta strands. Right? So this is quite easy to do it, like if you want to change your visualization of the molecule. And if you also notice here, there is this green colored molecule that is present in this particular structure. So this particular protein 6B2Q has a inhibitor bond to it. And this inhibitor is shown as green color here. It is not clearly visible right now, so I want to make sure that the ligand is clearly visible. So what I'm going to do is I'm going to click on the L option here. So you see that I'm showing a new button here. So I've discussed the style option. Now I'm clicking on the L option here, which is select my ligand molecule. Cell stands for ligand here. And so once I click on the L option, that will select my ligand molecule, which means that all the atoms of the ligand will be highlighted in blue color. If you remember when we tried to do 3D builder, we saw that the atoms were in blue color, the ones that you're trying to replace. In the same way, we're trying to change the representation for the molecule. Also, you're going to see that the atoms that for which you're trying to change the representation will also be highlighted in blue color if you have done the selection for those atoms. So now that I selected these atoms here, I want to make it visualized in a better way. So I'm going to go to style here, and the same style options is useful for trying to change the way your molecule is being shown in the workspace. We did explore these options when you're trying to build a simple ligand molecule because they are small enough, paracetamol was small enough. But when you have like protein with a drug, you want to make sure your drug is visualized properly in the workspace. So you go to style option. Now that these atoms are selected for the ligand, you can try to choose these options here in the second row. So by default, it shows your molecule in wire representation. So if you go to like thin tube here, it's going to make the ligand slightly bigger, still not perfect. You can make it into thick tube. It can make it like even bigger. You can show this a ballistic representation where every atom will be shown as a ball and the bonds of the atoms will be shown as sticks. Or you can show it as a CPK representation, which will show the whole molecule as spheres. Every atom will be shown as a sphere. So now you can clearly see where the ligand is interacting with the protein. And this is how the protein and the ligand are complex with each other in this particular case. All right. And also notice that, for this particular case, this is where the ligand is. And I want to see the binding pocket of the protein. So, right now I can see that, okay, this is where the ligand is, but how deep is the binding pocket? I have no information on the depth of the binding pocket. So if I want to find the depth of the binding pocket, I'm going to create a surface for the protein. So to create a surface for the protein, I just click on the P option here. So P stands for protein. If you select only the protein, it won't select the ligand, it only select the protein. So we did the L before, now we are doing the P here. And so once you select the protein here, you can see that all the atoms of the protein are blue color right now. Go to style and click on the surface option here, and that will generate the surface for your protein. So now you can see that this is your overall protein molecular surface here. With a few simple cases, you got the surface here. And you can see that this is the binding pocket into which the ligand binds to the protein. You can see how deep the pocket is and how well the ligand fits into the binding pocket very easily with this option here. Okay. So all I have done is I just clicked on the P option here, clicked on style here, clicked on surface here, which is the last option here, which will render this surface for you. Now, if you want to hide the surface, you can just, right now, I'm going to show you one more new button where you have on the bottom right here, you have this mesh here. So anytime you see something in blue color, that means that the molecule is selected or highlighted, or that option is highlighted here. So you can just like see that the surface toggle is right now in blue color, which indicates it's like it's being shown right now. If you click on the surface toggle, it will become green color, which will hide the surface for you. And similarly, you have a ribbon toggle here, which you can just like click on it. It's going to hide the ribbons also for you. But for these to be activated, you have to first create those ribbons or surface from this option here. And then you can turn them on or off, depending on what you want to turn on or off. Okay. I like to take a look at my protein in this way here. So I just like showed you this one. And so this is how you can try to like load your protein molecule. And if you want to take a further look at some information here, you can go to window here, click on sequence viewer, and that will display the sequence of the protein at the bottom here. So everything is like, you have so many widgets available to you here. It all depends on what you are trying to look at. Of course, there's only so much we can show you in a beginner introduction session, but the more you use the software, the more comfortable you'll become with it and all the options that are available in the interface. So this is a 1-letter amino acid sequence for the protein. I won't go into details here, but I'll just like clip this part for now. And so I have shown you this protein here. I've shown you this ligand here. And one more thing I want to point out before you go for the lunch break or the breakout session is that I have been trying to like, I've shown you, I've zoomed into the molecule, zoomed out the molecule, I have rotated the molecule. So how do you do these things? It's quite critical to be able to like, take a look at your molecule in 3D and see the different information that is available to you. So if you want to zoom in your molecule, if you have a mouse with you, not a trackpad of a laptop, but you have an actual mouse with you. So if you have a mouse with you, then the mouse will have a left click, a right click, and you have a scroll wheel on the mouse. That's how typical most mouses are. So you just like, if you roll the scroll wheel forward, that will zoom into the molecule. If you roll it backwards, that will zoom out of the molecule. So zoom in is roll the scroll wheel forwards, zoom out is roll it backwards. Now, if you want to rotate your molecule, you hold the scroll wheel, click on it. So click on the scroll wheel, hold the scroll wheel, and you move the mouse around. So that will rotate your molecule in whatever plane you want to rotate in. Okay, let me repeat that again. For zooming in and zooming out, you just roll the scroll wheel. You don't like click on it, you just roll it. But for rotating the molecule, you click on the scroll wheel, hold it, and then move the mouse around. This instruction is also provided in the handout, so you can just take a look at the handout later on if you want to find out how to rotate your molecule. And lastly, you have the left click and the right click that are left out. So right click is for like moving your molecule in any direction that you want to have. You can translate your molecule instead of rotating it. And left click will make sure that you can select a particular atom or in your workspace. You can see that when I click on this atom here, there's a blue color cube that appears on it, which means that atom is selected. Okay. So this is what I wanted to show you in this demo session. If you don't have a mouse with you, you can also use the keyboard shortcuts by going to view here. You can see that rotate will have the keyboard shortcuts of / or semicolon in the X direction, comma or full stop in the Y direction, and square brackets in the Z direction. Same thing for zoom in, zoom out. You have K as a shortcut keyword key for zooming in, and J as a shortcut key for zooming out. And you have shortcut keys for translate also. And let's assume that for some reason your molecule is like very much zoomed in. You want to like, get back to your original view. So for that, you can just click on this button with the 4 arrow marks here, which will fit your view to all visible objects. And that will make sure the view is like the whole molecule is brought into focus, what are you looking at? So if you just zoom in, you just click on this particular option here, and that will bring it back into focus. Or if you zoom out also, if you just like too small here, just click on it, will bring it back to focus again automatically for you. So with that, I'll stop this demo session. I know that the inauguration went a bit longer. That's why I had to like, be a bit fast with the demo sessions right now. But this is what we wanted to cover before lunch. And so post lunch, we're going to take a look at different calculations like preparing your ligand molecules for docking, preparing your protein and identifying the binding pocket, and doing the docking calculation and analyzing your docking results. All of those will be done post lunch session after 2 o'clock. And over to you, Dr. Prajwal.
Yep. Thank you very much, Dr. Kaushik. So, dear participant, just check the Zoom chat box. There are two handout links which I have shared. One is for building a small molecule using smiles as demonstrated by Kaushik. And second handout is for protein visualization. So please go through those handouts one by one and try to do the task exactly as mentioned in the handouts. And for that, I have opened the breakout room. Please do join the breakout rooms. And after this task get over, we will have the lunch break. So please do join the breakout room and let us know if you face any difficulty in doing that. So thank you very much. So welcome back, all of you. Sorry, it was a short breakout session, but no need to worry about it. You have handouts with you. Please give a try. Try to follow the handouts and do the task. And because we are already on time, it's 1 o'clock. So let's have a lunch break. But cloud instances are on, so don't worry about it. So you can use the cloud instances and try to do the task which is mentioned in the handouts. So both two handouts, try to follow it nicely, take your own time. And then we will all meet again at sharply 2 o'clock. And then we'll proceed with the workshop again. So the same Zoom link will be active again at 2 o'clock. So please do join at 2 o'clock. Take a look at Zoom chat box message. And please be on time. We have a lot of things to cover afterwards also. So it would be nice if you be on time. You will not miss the information. So thank you very much for joining it again so far. And then we will again meet at exactly 2 o'clock sharply. So thanks again and see you all. So here I just close this particular meeting. I just end it and then it will again active at 2 o'clock. So please do join through the same Zoom link. So thank you again. If you have any questions, we will discuss it after the lunch break. Thank you again. See you soon.